What is gene editing? Rewriting the code of life. Welcome back. I'm ADELINE Atlas, 11x published author, and this is the Designer Babies series, Reprogramming Humanity, One Genome at a Time. Let's strip it down. Before we talk about the ethics, the economics, the eugenics debates, or the billionaire-funded labs, we need to answer the core question. What exactly is gene editing? What does it mean to edit a human being? What are we doing when we cut DNA? How far can we go? And what does that process look like behind the sterile language of biotechnology? Because most people use the term casually. They'll say, "Oh, CRISPR? That's where you can change genes, right?" Yes, but it's deeper than that. Gene editing isn't just swapping out a paragraph in a novel. It's altering the master code of a living organism, before that organism ever breathes, speaks, or suffers. It's not theoretical anymore. It's happening in labs right now. To embryos, to sperm and eggs, to the cells that will become people. So today's episode is your foundational lens. We're not going to speculate. We're going to understand what gene editing is, how it works, and why it's more than science. It's authorship. DNA, the original operating system. Your body is built from instructions. Those instructions are encoded in DNA, deoxyribonucleic acid. Think of it as the original operating system, with over 3 billion letters of biological code per person. That code is made up of 4 chemical bases, A, T, C, and G. Arranged in specific sequences, they instruct your cells how to grow, develop, function, and interact. Genes are small segments of this DNA that hold blueprints for specific traits, height, eye color, metabolism, even aspects of behavior and risk for disease. Until recently, this code was untouchable. We could read it, we could study it, but we couldn't alter it with precision. Gene therapy existed, but it was clunky, unpredictable, and often dangerous. The dream of directly editing DNA, cleanly, safely, and with intent, remained out of reach. Then came CRISPR. What is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. That mouthful describes a pattern discovered in bacterial DNA, sections of code that store memories of viral invaders. Bacteria use these sequences to recognize and destroy viruses that try to attack them again. To do this, they use a protein called Cas9, which acts like a pair of molecular scissors. CRISPR tells Cas9 where to cut. Cas9 makes the incision. The virus is disabled, and the bacterial cell survives. What scientists realized is that this ancient bacterial defense mechanism could be hijacked and redirected. If you program CRISPR to recognize any DNA sequence, not just a virus, it will guide Cas9 to that location and make a cut. Once the cut is made, the cell's repair machinery kicks in, and that's the moment of opportunity. By controlling the cut and the repair, we can delete, replace, or insert genes. We can turn genes off, fix mutations, or even add new functions. And because CRISPR is relatively cheap and easy to program, it has democratized gene editing. What once required a billion-dollar lab can now be done by a graduate student, or even a hobbyist biohacker. This is the core breakthrough. We now have a search and edit function for the human genome. CRISPR is copy-paste at the cellular level, and the implications are enormous. Germline versus somatic editing. Temporary versus permanent gene editing comes in 2 forms, somatic and germline. Somatic editing happens in the body's cells after birth. For example, editing the genes of a person with cancer to make their immune cells more aggressive. This is medical therapy. It affects that person, and only that person. The edits don't get passed down. Germline editing is different. It involves editing sperm, eggs, or embryos. In other words, changing the DNA of a future person before they're even born. These edits are heritable. They are passed on to all future generations. Once a germline edit is made, it enters the bloodline. The change becomes part of human history. This is why germline editing is so controversial. It's not just medicine. It's modification of origin. You're not curing disease. You're designing life. and you're making permanent changes to the human genome with unknown ripple effects. This is what makes the designer-baby conversation urgent, because once a gene edit is in the germline, it cannot be undone. What can be edited? Thanks to advances in CRISPR and genomic sequencing, scientists can now identify and target thousands of genetic markers tied to traits and diseases. Some of the most common targets include monogenic disorders like cystic fibrosis, sickle cell anemia, and Tay-Sachs, immunity genes like CCR5, which was edited in the Chinese CRISPR twins to resist HIV, polygenic traits like height, IQ, and temperament, though much more complex. Appearance genes, including those linked to skin tone, eye color, and hair texture. Behavioral and psychological predispositions, including risk for depression, ADHD, or even autism. What's critical to understand is that some traits are governed by a single gene, while others, like intelligence or empathy, are influenced by hundreds or thousands of genes working in concert. Editing polygenic traits is far more complex and risky, but that hasn't stopped companies from offering polygenic screening of embryos to help parents choose the best combination of traits during IVF. This is not full editing, but it's selection, and selection is a form of design. Limitations and dangers. Gene editing may sound clean, but it's far from perfect. There are major concerns, including off-target effects. CRISPR sometimes cuts in the wrong place, leading to unintended mutations. Mosaicism. Not all cells may carry the edit, leading to mixed results within one body. Pleiotropy. One gene often affects multiple traits. Editing for intelligence might also increase risk for anxiety or cancer. Ethical ambiguity. Where's the line between enhancement and coercion? Between freedom and manipulation? The more we try to control biology, the more we learn that genes don't exist in isolation. The genome is a web. Pull one thread and the whole system shifts. What seems like an upgrade in one generation could be a vulnerability in the next. And yet, the industry moves forward, because even with these risks, the demand for genetic control is growing. The rise of direct-to-consumer genetics gene editing doesn't live in secret labs anymore. Direct-to-consumer companies like Genomic Prediction, Orchid Health, and others are now marketing embryo selection and risk scoring directly to prospective parents. You send in your sperm or eggs. They run your DNA through their proprietary algorithm. You receive a report: projected IQ range, projected height, disease risk, emotional profile. From there, you can choose which embryo to implant. In short, you're now shopping for a child, based on predicted genetics. These companies use terms like empowered parenting and advanced family planning. But what's really being sold is control, prediction, insurance against imperfection, a guarantee that you've done your best to create the best possible child. But prediction is not destiny. Genes are not fate. And no algorithm can measure global legal landscape. The legality of gene editing varies wildly by country. United States. No federal law bans germline editing, but it's barred from receiving federal funding. Private clinics can operate in gray areas. United Kingdom allows mitochondrial editing, 3-parent babies, but bans germline CRISPR. China officially prohibits editing embryos for reproduction, but enforcement is inconsistent. Russia, loosening restrictions. European Union, strong bans on inheritable edits. Middle East and Africa, wide variation with emerging biotech investment. In other words, there is no global consensus, and wherever a law prohibits, someone will work around it. This means the future of human evolution may not be determined by morality or science, but by jurisdictional arbitrage. Editing ourselves into a corner? The promise of gene editing is seductive. Eliminate disease, extend life, perfect humanity. But what we edit out today may be what we needed tomorrow. Nature evolves through diversity, mutation, and randomness. Our resilience is often forged through suffering. Our gifts sometimes arrive hidden inside our flaws. If we edit too much, too soon, or too blindly, we risk creating a brittle biology, one optimized for convenience, not strength, one vulnerable to unknown pathogens, unable to adapt to harsh environments, and disconnected from the ancient chaos that made us human in the first place. This is the paradox. The more we perfect, the more we weaken. The more control we demand, the less resilience we build. And in trying to create better humans, we may be writing the end of what made us human at all. Gene editing is not evil. CRISPR is not demonic. These are tools. Tools that hold both miracle and mayhem in their grip. Tools that can heal and harm, liberate and enslave, depending on the hands that wield them. The issue is not whether gene editing should exist. It does. The issue is how we use it, who controls it, and what we forget in our pursuit of perfection. Because once we begin to rewrite the human script, we must ask, who becomes the editor? And what happens when the next generation is not born, but built?